Thanks a lot, Tim. It's a pleasure to be here at the Allen Institute. Tim didn't mention that he was my graduate student at Northwestern, so I don't know why he left that part out. I'm going to tell you today about a couple of projects from the lab. One of them was published last year, and the other one is on bioRxiv, but not yet fully published in final form. But before I do that, I'd like to actually just take a few minutes to talk about what motivates me, what kinds of things we're interested in, and how we choose the problems that we work on in the lab. And I think for many of you here, none of this will come as any surprise because we probably have a shared philosophical view of neuroscience and maybe biology in general. But I nevertheless hope that you find this sort of background useful when you think about the projects that we're doing in the lab, and maybe it'll make for some interesting conversations later on. So ever since I started in neuroscience, which was more than 30 years ago now, I've been interested in memory. And in fact, it was my interest in memory that motivated me to go into neuroscience in the first place. And that interest just really derived from introspection and thinking about the notion that it's quite amazing that we can remember things that happened a long time ago. So sometimes a single event in our life history can be stored in the form of a memory that lasts a lifetime. And I can think of many examples from my own life history where I can think of things that happened many decades ago, and I can recall those events pretty vividly. And I think it's an amazing feat of biology that we can do that, and I would like to understand how that occurs. So to do that, I have relied on studying the hippocampus because of the well-known role of the hippocampus, which is shown here in a human brain and here in a rodent brain. In humans, it has a well-known role in declarative memory. In animals like rodents, obviously, we study those for practical reasons. They don't really have declarative memories, or at least we can't ask them about their declarative memories, so we have to infer the kinds of memories that they have from their behavior. And probably most of you are familiar with the kinds of tasks that are traditionally used in the field to assess whether an animal has formed a memory or not, and whether the memory, how long the memory lasts, for instance. So, what we would like to do is to try to understand the neurobiological underpinnings of the memory formation. And more specifically, I think we should probably talk about memory-guided behavior. And in rodents that we study, typical memory-guided behavior is a navigation task. Those are very relevant to rodents because the hippocampus is believed to have evolved hundreds of millions of years ago, probably to assist animals in doing intelligent navigation. It's one of the first intelligent things you might imagine an animal evolving to do, is to go find food and avoid danger and come back to a safe place. So that's the thinking, is the hippocampus evolved to support intelligent navigation. We want to understand how that works. But of course, the hippocampus doesn't do that on its own. It sits inside the brain, and it communicates with many other parts of the brain. So it's part of a very, very complicated circuit that drives memory-guided navigation. So that raises the question, what does it even mean to understand something like memory-guided navigation? And I'll touch on that a little bit. One of the criticisms that some people have of studying something like memory in animals is they say, well, maybe monkeys are smart, but mice are really not that smart. And I disagree with that assertion completely. I think mice are very smart. They've survived evolutionarily for millions of years. They have to be smart to do that. And just as a fun example of how smart animals are, I recorded this video. So recently, my wife's parents moved from Texas to Virginia to be closer to us, and they installed this bird feeder in their backyard. And the bird feeder is designed so that squirrels can't get into it. So maybe you can predict where this is going. This bird feeder has a bar, and when a bird stands on it, it's so light it doesn't matter. It can access the seeds, but if a squirrel rests its paws on this bar, it closes it so there's no access to the seeds. Well, the squirrels quickly figured out how to get around this little contraption. And you can see that the squirrel is suspending itself from its hind legs and accessing the seeds by removing its weight transiently from the bar so it has access to.
Oh my goodness.
Should turn off the sound, I guess. We don't really need that. But if you ask me, this is pretty intelligent behavior. I mean, a bird feeder like this is not something that the animal evolved to outsmart. It evolved to, really have intelligent capabilities that allow it to adapt to new situations all the time. So I think, it's not a mouse, it's a squirrel, but still, I think these are the examples of really smart things that animals can do. And in fact, I recently started reading this book that's called Are We Smart Enough to Know How Smart Animals Are? And there are many examples in this book of the intelligence of different types of animals, and I think it's worth a fun read, probably of interest to almost any biologist. And the question is, are we smart enough to understand how smart animals are? Can we understand something as complex as the neurobiological underpinnings of memory-guided navigation in mice, for instance? I don't know the answer to that question. I don't know if we can or can't, but we certainly are going to keep trying. And to highlight or illustrate how complicated this problem is, consider the rapid advances that we've been experiencing lately in artificial intelligence systems. So this is a paper from a paper that was published last year. The DeepMind group in London designed an artificial intelligence system wherein an agent can learn to navigate a pretty complicated environment to get to a goal, from a start site to a goal. Initially, some of these doors are closed, and then after some training, the animal learns to navigate in this thing, and as soon as the door opens, the animal starts using this path to shortcut to the goal. So that's pretty intelligent behavior of an artificial agent. And interestingly, the DeepMind team showed that if you enforce conditions in this artificial intelligence system such that they have a representation that mirrors that of grid cells in the mammalian entorhinal cortex, these agents outperform systems that don't have grid cell representation. So that's kind of an interesting insight into the potential importance of grid cells. But nevertheless, if you talk to the people from DeepMind who designed these systems, they don't claim to understand how it works. And it's an interesting question. What does it even mean to understand how something this complicated works? And these are intelligent but very complex artificial intelligence systems. They have different modules. Each of these modules consists of many layers of, quote-unquote neurons connected to each other by adjustable weights. They're tuned for experience to minimize some objective function. There are different modules with different numbers of cells and different layers. They're very complicated systems that these engineers have designed. And each one of these modules can be very complicated, many, many layers. This is another example of a network that was designed to play the game Go. These agents are now really, really good at these complex tasks, but the people who engineered them, the very people who built them, don't claim to understand how they work. Now, furthermore, there are certain things that animals can do that these artificially intelligent systems cannot do. For instance, animals can learn things through one-trial learning. They can learn to switch the valence of a particular reward from something that is, something, for example, that's very aversive to an animal, under a new set of conditions can instantly become very attractive to the same animal. Artificial intelligence systems can't yet solve that problem of, sort of almost instantaneous switch from an aversive to an attractive stimulus. So there are many examples like that of things that biological systems can do that artificial systems can't yet do. And then furthermore, as neuroscientists, I think we all really want to do more than just build things that reproduce the behavior of animals. We want to understand how animals actually really do it. What's the biology behind it? And for instance, the neurons in artificial intelligence systems look like this. They have some input thing you might think of as a cell body and some output thing you might think of as a synapse with adjustable weights. But it was pointed out to me one time by Andrew Welchman that this looks kind of about as much like a neuron as that looks like a person. And of course, real neurons are much, much more interesting structurally and functionally. And I, for one, would like to understand in detail how neurons like these 3 different types of neurons in the hippocampus, how they function. Why is it that in different parts of the hippocampus, the structural and functional properties of the neurons are different? I think these are important questions that we would like to answer along the way towards, on this path towards understanding something as complicated as memory. Okay, so how are we going to do this? As a group of neuroscientists, we choose typically to study the system at some particular level, molecular, cellular systems, or behavioral and cognitive neuroscience. And I think we probably all agree that a major goal of neuroscience in general is to link or connect our understanding of neuroscience at these different levels. So for example, we would like to understand the function of cells in terms of their molecular properties, and we would like to understand systems in terms of the cellular composition of those systems. So this is what I think it means to come up with mechanisms, is to go back up one level and say, okay, we observe something at the systems level. Can we explain, based on the cellular architecture or even the functional properties of those cells, how that observation emerges? And to do that, we like to build models. We make experimental observations, and then we like to build models of the system, do experiments to challenge whether those models really make sense, and as Tim pointed out, then refine and update the models based on more and more observations. Ideally, I would argue that we will need computational models of these connections between levels to really be able to claim that we understand them, because if we can't build computational models based on the ideas that we have in our head, one might challenge whether those ideas in our head really make as much sense as we think they do. So I think overall, this is what it's going to mean to understand something like memory-guided navigation in a rodent, is can we go all the way from molecular to cellular to systems to the behavior and the cognition that underpins that, those intelligent behaviors. So that, I would say, is a summary of the overall goal. As Tim mentioned, I'm now doing this. My lab is at the Genelia Research Campus in Northern Virginia, just outside of DC. This is an amazing place to work. I was saying to Tim that this week, by being here and having been at Genelia at home a couple of days ago, and I'm going from here to the Sainsbury Wellcome Center in London, I think in 1 week I'll have been in the 3 coolest buildings in neuroscience in the world. So I think Genelia is a cool building. But more importantly, the people are amazing. The structure of the way we do science there is amazing. And I'm happy to talk to any of you who are interested about how things work at Genelia. This is my lab. Actually, a couple of the people on this slide have now left the lab. But I'm going to talk a lot today about work that was done by Mark Zembrowski, Xinyu Zhao, a little bit of work from Qinglong Xu. And I just regret that I can't talk about everything that we're doing in the lab, and all these other folks have done some really exciting and cool things. Now, one of the things I want to point out is that we have this highly collaborative model at Genelia. So even though I just mentioned 3 people whose work I'm going to highlight today, this is a full list of all the people at Genelia who've contributed to the work that's going on in the lab. So it's really, even though it's a fairly small lab, we benefit from the efforts of lots and lots of people. So I'm very thankful for that. Okay, and then I'm also going to mention briefly the work of a project team at Genelia called the Mouse Light team. Project teams are groups of people that are assembled to do a project that is of interest to multiple labs at Genelia, and you'll see some of the results from this team of people. Okay, so memory-guided navigation in the mouse, in part, is determined by the function of the hippocampus. And I'm going to talk today quite extensively, actually, in this next part, about the subiculum, which is just posterior to the CA1 region of the hippocampus. It is the major, or one of the 2 major output structures of the hippocampal formation. And as you'll see, the neurons in this part of the brain project to many different targets outside the brain. And we've done a sort of an assessment of the cell types in the subiculum using single-cell RNA-seq approaches. This is work from Mark Zembrowski in my lab. And the basic idea here is to microdissect the subiculum, dissociate the cells, and then do single-cell RNA-seq. All the data you can find for subiculum and other hippocampal cell types at this website called HippoSeq. And we've used the single-cell approach to do cluster analysis of the cell types in the subiculum along that full axis going from the dorsal to the ventral part of the brain and in other dimensions that I'll talk about as well. Okay, so the results, just to quickly summarize them for you, look like this in a t-SNE plot. And you can see that there are 9 different clusters here. It turns out that one of these clusters, I believe it's this dark blue one here, is actually CA1 cells. So there are 8 different types of cells, roughly, by this type of single-cell RNA-seq assessment of the cells in the subiculum. Mark used, actually, resources generated here at the Allen Institute to describe the domains in which these different genes are expressed in the subiculum. So you can map these different colors and marker genes that correlate with these different clusters over here to their location in the subiculum through the Allen Institute Hybridization Atlas. So this was a super useful resource for us. And this is sort of, I would say, a draft version of the cell types and their location in the subiculum. And one of the things we'd like to understand is what are the functional properties, the projection targets, et cetera, and the role in memory-guided navigation of these different cell types. Okay, so the mouse light team has helped us in our understanding of the projection classes. So I'm going to show you a little video here. I don't have time to talk in detail about the mouse light team and the approach that they use to doing the imaging and reconstruction of single cells. These are examples actually from pyramidal neurons in the motor cortex. This is cell types that are of interest to Carl Svoboda's lab. And you can see these really gorgeous and elaborate axonal arborizations of individual neurons. This is from the thalamus. So we've been doing this, the mouse light team has been doing this across the mouse brain. They've now reconstructed on the order of 1,300 neurons. A paper was published last week describing the first 1,000 neurons that were reconstructed. They're up to 1,300 or so now. And we're just going to keep going and hoping to collaborate with Hong Kui and others here on doing more of this. So this team has helped us in assessing the different types of neurons in the subiculum and their projections from the subiculum to other parts of the brain. So I'll let this video finish just because the finale is kind of fun, and then we'll move to the subiculum projections. Okay, so you can read all about, you can read all about the mouse light team in their recent paper, for example. Okay, so we've done these single cell reconstructions for the subiculum, and specifically for cells labeled in the dorsal part of the subiculum. And it turns out there are several cell types in the dorsal part of the subiculum. 3 of them are illustrated here. And in the next part of the talk, I'm going to highlight the function of 2 of them. One of them is shown in magenta here. And this is a class of cells that projects to the retrosplenial cortex. And another cell type is shown in green here. And this projects to the nucleus accumbens, among other places. So most of these neurons don't project to just one place, but to multiple places. That's something new that we learned from this effort. Why is this going here again? Okay, so I actually want to go back. Let this thing. Okay, so I just want to show you the 4 different classes of subiculum neurons that we identified based on their projections. So one of them is something we call those cells having broad projections. They project to many different parts of the brain. The contrast to that is cells that have almost entirely local connections within the subiculum. And in one case here, and there's a couple of other examples, there's one axon that goes out and sort of ends somewhere, but it doesn't really elaborate. And this is maybe a functional connection, but maybe not. It looks mostly like most of its synaptic connections are local here within the subiculum. There's another class that projects to the thalamus as well as local connections. Another class that projects to the hypothalamus as well as the retrosplenial cortex. So we came up with these 4 descriptions. If I can talk to you offline about how we decided there were 4 types and not 5 or 6 or 7 different types. I think this is potentially a little bit arbitrary, but it certainly looks, looking at the data by eye, like these 4 types make sense. And we can map then these 2 cell types, at least that I talked about, the one shown in magenta here and the one shown in green here, onto these different clusters from the single-cell RNA-seq analysis. The colors don't match, so the magenta cells that project to retrosplenocortex, they're over here in Alaska. It's amazing that this looks like a map of the world somehow. India is missing, South America is missing, I don't know, but it kind of looks like the world. These cells that project to nucleus accumbens are mostly in this group of cells I call Canada. Some of them are over here in Scandinavia. So we're beginning to understand the correspondence between cell types based on gene expression and cell types based on projections of the axons. But that effort is definitely far from complete. We need to do a lot more work to have a full picture as far as mapping gene expression onto morphological classes. But we do have, quite a bit of information, enough that I think, that we were able to begin to start asking questions that would allow us to make these connections between the different levels that I talked about. And our goal really was, okay, 8 years ago when I started at Genelia, we already understood that there were at least 2 different cell types in the dorsal subiculum. And we thought, wouldn't it be cool if we could determine whether these 2 different cell types in the dorsal subiculum have differential contributions to memory-guided navigation or memory in rodents. And in order to do that, we're going to have to be able to go from the cell types to their molecular identity so that we can manipulate these cell types based on our knowledge of their gene expression and examine the consequences for behavior. Okay, so that last step that I just described is going all the way from cells based on molecular properties, gene expression specifically, to their role in behavior. And that's, I'm going to show you what we learned from that, and I think it's cool. But really, to understand it, I think we're ultimately going to have to be able to make these connections step by step instead of jumping from molecular properties to behavior. Okay, nonetheless, it's sort of proof of principle, I think, that we have the tools that we need to do to be able to do this. So, okay, here's a picture of the hippocampus. Probably you've all seen this before. I'm not going to go through the connectivity on this diagram. Instead, I want to schematize the whole thing. And the basic architecture is that cells in the entorhinal cortex project to the hippocampus. And the cells in the hippocampus project back to the entorhinal cortex in a big loop, as well as outside of the hippocampus, which I'll show you in a moment. So, for example, entorhinal cortex projects to the dentate gyrus, to CA3, to CA1, to subiculum, and then out to back to entorhinal cortex and out to other places. And there's all kinds of loops. You can see these loops all over the place. There's recurrent loops in CA3. You can imagine a loop where going from entorhinal cortex to CA3 to CA1 and back to the entorhinal cortex. There's recurrent loops all over the place. This is sort of a general feature of these neural circuits, which I think will be important, and I'll bring that up in another context in a moment. Okay, so Mark Zembrowski really led this effort, as I mentioned, to study cell types in the subiculum. He led the effort on the single-cell RNA-seq approach, and it was his goal to then be able to silence these cells in behaving animals. Okay, so I told you that subiculum, as well as CA1, project out to a long list of targets outside of the hippocampus. And it turns out that there's some interesting sub-architecture that's important for this structure, this part of the talk, and that is that there are differences in the cell types along what we call the proximal distal axis of the subiculum. So there's 2 cell types that I showed you in the reconstructions. One of them is in the proximal part of subiculum, and one is in the distal part of subiculum. And there's a similarly functional gradient along CA1 in this proximal distal axis. Okay, so we knew this before, that there were at least a couple different cell types on this axis in rats, and Jim Knierim's work at Hopkins has given us a lot of information about what the potential functional differences are in behaving animals in these cell types, or these location-dependent properties of the cells. And others have done sort of circuit work, and based on this work, we have this idea that there are 2 parallel circuits in the entorhinal cortex to CA1 to subiculum loop. One of them goes from medial entorhinal cortex to proximal CA1 to distal subiculum and back to medial entorhinal cortex. The other one goes from lateral entorhinal cortex to distal CA1 to proximal subiculum and back to lateral entorhinal cortex. In addition, we know that these two regional subdivisions, which are shown in these two different colors, project to different targets outside of the hippocampus. And we can take advantage of this now to use retrograde labeling techniques, for example, putting retrograde tracers in retrosplenial cortex or the ventral hypothalamus labels distal subiculum cells, nucleus accumbens or prefrontal cortex labels the proximal subiculum cells. And an example of that is shown here. This is distal subiculum. You can see from the inset, here's CA1, here's distal subiculum, proximal subiculum. So distal is labeled with a retrograde injection of a tracer, retrograde virus tracer in the prefrontal cortex, and this is a nucleus accumbens. And so you can see that there's a clear boundary separating these two different types of cells. And as I mentioned, we know something about gene expression of these two different types of cells. And so we have now access to these two populations of cells in distal and proximal subiculum, some understanding of where they project to, and some understanding of the different types of genes they express. I should also say that we have done electrophysiology on these two cell types and identified clear differences. And our previous work in the rat has actually shown that there are differences even in things like the plasticity rules for these two different types of cells. Okay, so we have now this sort of distal proximal distal axis of the subiculum, dorsal subiculum in particular. Differences we know in the outputs. We also know there are differences in the inputs to these different regions and cell types, and differences in the firing phenotypes. So to really get molecular access to these two cell types, Mark did another RNA-seq experiment involving labeling of the cells using retrograde beads. So he injected retrograde beads of one color into nucleus accumbens, another color into retrosplenial cortex, and over here you can see the two cell types labeled with this clear boundary between them. Then we could sort these cells based on the color of the beads that they express and do population-based RNA-seq analysis of the two populations of neurons. And, okay, yeah, so if we now plot gene expression for these two populations, so the idea is for each gene is a point on this plot. You've got one population here plotted against the expression value for the other population. All the points in black are roughly equal gene expression levels for those genes, and all the colored points are genes that are expressed at different levels using a 5% false discovery rate as the threshold. And you can see that there are hundreds of genes that are enriched in one or the other population of these cells. So there are clear, in addition to the location of these cells and their projections, which we know there are clear differences in gene expression. If we look at the types of genes that are differentially expressed, many of them have phenotypes that are associated with neurons, things like axon guidance and adhesion molecules. Obviously, those might determine where those cells are actually projecting in the brain during development, which types of synaptic partners they find when they get to the target region. There are differences in peptide and receptor expression, synaptic proteins, voltage-gated channels, and so on. So there are clear gene expression differences that not only serve as markers for these two populations of cells, but also likely are responsible for the differences in their functional properties. Okay, we validated the differences in gene expression identified from RNA-seq by doing FISH. So we didn't do this for all the hundreds of differentially expressed genes, but just for some of them. Here's a couple of examples here. You can see, again, this boundary between the two regions based on expression of these two genes, doing two-color FISH experiments. And so now we can add to the list that we have clear differences in gene expression for these two regions and cell types in the subiculum. Okay, so now we're in shape, right? We have marker genes for these two cell types, and we can take advantage of this to try to silence them in behaving animals. So here's how the experiment was designed. We found a Cre line, this click8 Cre line, that selectively expresses this gene CLIC8 and therefore drives Cre expression in proximal subiculum. And you can see this because if we inject a virus carrying a flexed version of mCherry into this part of the proximal part of the subiculum, you can see selective expression in proximal subiculum. And we identified distal subiculum with a retrograde viral tracer in this case, in the hypothalamus. And you can see the non-overlapping expression of these two color markers, okay? So the reason that these two colors are non-overlapping is not because the virus is spatially restricted to the proximal subiculum. And we targeted that region, but undoubtedly it leaks over into distal subiculum as well. It's just that the cells in the distal subiculum are not expressing Cre, and therefore they don't express the reporter gene, okay? We can do the complementary experiment using another Cre line, this one for neurotensin, and the complementary experiment targeting the flexed virus here to the distal subiculum, getting accordingly expression of the marker in the distal subiculum, and then retrograde tracer in nucleus accumbens, again, non-overlapping expression. Okay, so we can do the experiment of selectively driving transgene expression in one population of cells or the other using these two different Cre lines. And if you're paying close attention, you probably notice it's not just an mCherry reporter, but it's also HM4D expression. So HM4D is an inhibitory dread receptor. And if you then give the animal an injection of the agonist for that receptor, which is CNO, it causes silencing of these neurons. And this doesn't always work in all systems. It depends probably on differences in neuronal phenotypes and the connectivity, recurrent connectivity of the circuit. But we validated that this actually works quite well in these two areas where we've targeted the gene expression. So we did recordings to show that when we give animals CNO, the firing is dramatically reduced in these regions in vivo. Okay, so now we're positioned to do behavioral assays with one or the other of these two populations of cells silenced. And Mark did that, got no effect on locomotion, no effect on anxiety-related behavior, object recognition, olfaction, but he did get a very interesting phenotype on the Morris water maze test. So probably most of you have seen the Morris water maze. It's a very good assay for memory-guided navigation in rodents. You put them into this cloudy pool of water, and they're very good swimmers, but they know they can't swim forever, so they're looking for a way out. And they actually know, because they've been trained, that somewhere in this environment there's going to be a platform that they can stand on. And they swim around looking for the platform, and this guy is eventually going to find the platform pretty soon. And once he finds it, he gets on and stays there, okay? Now, if you do this repeatedly, like 3 or 4 times in one session, they learn and remember the location of the platform. So here's another animal. This animal has already been trained by 3 prior introductions into the pool. And as soon as you put him in there, he swims straight to the remembered platform location, okay? So this is a very good assay. It turns out that if you take the same animal and the next day put it into the pool, it will swim to the location that it learned on the previous day. Okay, so they have very good memory of this, and it probably works well because their life depends on it. It doesn't really depend on it, but they maybe think it does. Okay, so we did a version of this assay where every day we do a session of training, and we change the location of the platform every day. And every single day, the animal learns within 3 or 4 trials it can reliably swim straight to the hidden platform. And then on day 10, we do a test where we inject the animal with either CNO or a control vehicle injection. But this time, we don't put the platform there, okay? So you're assessing whether the animal remembers the platform location. So first you train the animal on this location, and then you remove the platform. And this probe trial is an assessment of whether the animal remembers or has learned the location on day 10, okay? And if they do remember it, they swim around looking here, not all the time, but most of the time, okay? Then on day 11, you do the complementary injection of vehicle or CNO, and you do the same thing, okay? And here's the results. So these are control experiments. In control experiments, there's no silencing. And you can see on the probe tests, the animal spends most of its time in the target quadrant. That is where it has learned the location of the platform. Here's another one, okay? They don't completely perseverate. They'll go look other places, but they keep coming back to that location, kind of like you keep going back to where you think your car keys should be. Now here's the silencing results. If we silence proximal subiculum, there's really no effect. But if we silence distal subiculum, there's a very striking effect. And that is the animal does not remember or has not learned the location of the platform on day 10 or day 11, depending on which day it got CNO. And here are the summary data for this. This is distal silenced. It behaves at sort of chance performance on the amount of time it spends in the target quadrant. Okay, so this is kind of cool, but it's actually much more cool than this because we can learn more about the behavioral deficit or the memory deficit from these experiments. And that's because if you look at the performance on any given day, the animal on its first trial, that is the first time you put it into the pool, I already told you this, it will swim around in the location of the platform on the previous day. So it learned some location on the previous day. And now the question is, when you silence distal subiculum with CNO, does the animal actually do this or not? Okay, so you've got this pre-CNO session. The animal learned the platform location. Now you give it a CNO injection. And if it can't remember the location from the previous day, that suggests a retrieval impairment because you know it learned it on the previous day and it's unable to retrieve that memory, okay? On the other hand, if it does remember the location from the previous day, then its retrieval is intact, but on the following day after the CNO silencing, and by then the CNO has worn off, if it can't do this, then it's been an encoding deficit. And the results of this were very clear that retrieval of the memory was intact, but encoding was impaired. So what does this mean again? It means that you silence the output of the distal subiculum, and animals can still remember and use their memory of the location of the platform on the previous day, but they can't form a memory of the new location on the day when that part of the subiculum is silenced. Now, here's the summary data that support that. This is all published last year. Okay, so the reason this result was surprising to me is I expected the opposite, actually. Because, as I mentioned, subiculum is an output structure from the hippocampus. Many, many studies, over the course of decades have studied synaptic plasticity and, sort of, I would say, shown that synaptic plasticity contributes to memory formation in these parts of the circuit, dentate gyrus, CA3, CA1. And so my working model of all this is that memories are formed in this part of the circuit, and that whatever information is stored there goes out to other parts of the brain through the subiculum, and that's how memories are retrieved and used for the animal to guide its navigation. That was my assumption. That appears to be incorrect, because if we silence the output of subiculum from distal subiculum to these structures and others, and back to the medial entorhinal cortex, it prevents memory formation. So what that tells us is that communication of the hippocampus with other parts of the brain is essential for memories to be formed in the first place. And that, I think, is a change, at least for me, it's a change in the way I think about how memories are formed in the brain, in mouse brains. Okay, so follow-up experiments. I mean, what we did essentially is we inhibited these outputs from the subiculum. We could obviously, silence one or the other instead of doing both of them. So those would be follow-up experiments. Another thing is we could change the timing of when we silence them. So maybe if we let the animal form the memory under normal conditions, and then as soon as the memory is formed, we give it the CNO injections, maybe it inhibits consolidation of the memory. So I think there are many follow-ups we could do to make more progress on this. Okay, so now we got behavioral contributions. this is exciting to me, but at the same time it's sort of discouraging because this is all very slow. It takes a long time, and I think it's going to take us really a very, very long time before we have a good understanding of how these cell types and the circuits that they participate in actually contribute to memory formation and memory use. Okay, I'm going to use the last 10 minutes or so to quickly summarize another project, which is done by Xinyu Zhao with a collaborative project with Jeff McGee's lab, and Yingshui Wang from Jeff's lab contributed to this, as well as more recently, since we put the paper on bioRxiv, there's been a contribution from another postdoc in the lab. And that project involves recording from cells in the hippocampus in animals behaving in a virtual reality environment. And the goal here is that we would like to do memory-guided navigational experiments in head-fixed animals so that we can image from large populations of neurons, record from individual cells with whole cell recording, and do these kinds of circuit manipulations that I talked about in more controlled conditions where we can assess not only the behavior of the animal, but also how it affects the function of the circuit. But for now, just to get us started on this, we're just recording whole cell in CA1 pyramidal neurons in the hippocampus in these animals, head-fixed, behaving in VR. And they're doing a very simple task, which is they're running around an environment, in this case, a virtual oval, and there are different cues on the walls at different positions, and they're collecting rewards as they run around. And it looks like this. There's the animal, head-fixed, running in VR, and there's what the animal sees on the screens in front of it. And you can see the different cues on the wall. Okay, so because it's head fixed, the animal's stable enough that we can introduce a whole cell recording electrode and do a whole cell recording from a CA1 cell while the animal's running in this VR environment. And nothing's taken out. We just go through it, go through all of it with outward pressure. Okay, so here's a schematic depiction of the environment. These A, B, C, D, E, F refer to different cues on the wall. And these blue and orange regions are going to be relevant, as you'll see in a moment. Okay, so when you record from a cell, typically or commonly, this is the membrane potential as a function of time. On lap 1 around this environment, and lap 2, there's nothing. There's no firing. There's no synaptic potential, and you might think, how do you guys even know this is a cell? Well, we can make it fire, and later on we can reconstruct it and see that we actually recorded from a cell, but they're silent. They're not doing anything. But fortunately, we can take advantage of a trick that was developed by Katie Bittner when she was a postdoc in Jeff McGee's lab, and this is called behavioral timescale synaptic plasticity. So here's the trick. You inject current into the cell. It's like a large current injection that drives firing of this cell, and in fact drives like a large plateau potential at some location. So we did it when the animal was at this location marked by the blue lines here with QA. And you can see that when you do this once, even on the very next lap, the cell starts firing at that location. And we do this a few more times, but you can see that the cell, even after you stop injecting current over these 4 laps, the cell now robustly fires at this location. So what do? We created a place cell from scratch, basically. And a lot of work from Katie and Jeff has shown that the reason this works is even though the membrane potential is flat out here, it's not getting no synaptic input. It's getting some synaptic input everywhere. And you selectively potentiate all the synaptic input that the cell is receiving at this location, okay? And then that produces this robust increase in the ramp depolarization. It causes firing, and you can also see increase in the theta amplitude of the theta modulation of the membrane potential. So here's the current injection site, and you can see this huge change in the ramp depolarization is shown here in blue after filtering away the theta and the spikes. You can see this ramp depolarization around the current injection site. It's all a result of synaptic plasticity, and it's a single shot synaptic plasticity. It really only requires 1 time. We do 4 to make it more robust, but it happens even after the first shot. Okay, so we asked the question, what happens what is the cell responding to in this VR environment? We think of hippocampal neurons as place cells, but what is it about the place that they're in that's causing them to fire that makes them fire? So we said, well, what if we take this cue that the animal can see when we created this place cell, and we just move the cue to another location? So we replace D with A. And when we do that, you can see there's some firing here. So the animal's in a different place now, but it's responding to the same visual stimulus that it saw when we induced the plasticity. However, if you look at the group data across all these different measures, ramp depolarization, theta amplitude, and spiking, in the new location, it's weaker than it is in the original location. Okay, so you've sort of partially reproduced this firing in this by moving this visual stimulus. And we thought, well, is that because A following F is different from A following C? Or is it because A with B out in front of it is different from A with E out in front of it? So we tested this by just putting a virtual curtain, if you will, like a black curtain that the animal can run right through, so that when we induce plasticity, the only thing the animal can see at this point is this QA. And now we're going to ask, what if we move it over here? Okay? And here's the answer. We get much more robust creation of firing at this new location if the plasticity was induced when the animal could only see QA. And here's the group data over here. Okay? So this is actually an amazing thing, right? You take a place cell that fires in this location, and now you can make it fire in 2 locations. Or we can ask, well, what happens if we remove QA? First, we induce plasticity here at this location where QA is located. Then we duplicate it and remove it at this original location. And now we move the location of the firing of this neuron. So the neuron is essentially firing every time it sees QA, and it requires this visual QA in order for it to fire. Then it's necessary and sufficient. Which sounds really weird, right? Because now it just sounds like a neuron in the visual cortex is just responding to a visual cue. But we actually had trained the animals to run in 2 environments. The second environment is different in many ways. It's a different shape. The animal has to run in a different way. The cues on the walls are all different. And now we can ask, well, what if we present this QA, which we know is driving firing, right? And we introduce the plasticity in this environment. Then we teleport the animal into this other environment and put QA there. And the answer is, in this new context, QA doesn't do anything. Okay? So this is also an amazing effect, right? This cell went from responding to A and only A, essentially, to not responding to A at all just because it was happening in a different context, in a different virtual environment. And we already knew, shockingly, I think, okay, let's use this one, that it didn't matter what happened before or what happened after QA. All that mattered was QA in this environment. Now you put it in this other environment, and now QA doesn't do anything. So this means that there's some complete contextual gating of the cell's response to a visual stimulus that depends on the environment that it has placed itself in. I would argue that the animal knows when it's teleported into this new environment, oh, I'm in a different place now, okay? And it remaps, it reconfigures the whole hippocampus and maybe other parts of the brain as well. Hey, this is a very robust effect. Here's the group data. There's no effect when you introduce this cue in the second context, as you can see in the red lines here. Okay, so how do we interpret this at the cellular and synaptic level? One model you could imagine is that there's a sensory cue that arrives in the form of synaptic input to this cell and separate cues for context 1 and context 2. I put them on different positions on the dendrites. It doesn't mean anything. It's just so you can visually separate them easily. Okay, so one possibility is you've got some input associated with the context 1, some input associated with the sensory, producing some synaptic depolarization of the cell. Context 2 is not active, so you get nothing. The cell is summing up these inputs and depolarizing. It's all way below threshold, so we essentially see nothing. Okay? Now we induce plasticity. We potentiate all of these synapses, context 1 and sensory input. They get big. The cell now fires. Okay? the prediction of this model is that when you move the cell into context 2, so now these cells are going to be, although they've been potentiated, these synapses have been potentiated, these cells will be silent. These cells have not been potentiated, but they're still active. And these potentiated synapses should produce some synaptic depolarization. So, there's this condition where these potentiated synapses are active, these potentiated synapses are silent, these synapses are weak, and you get a sub-threshold depolarization. But that is not what we see at all, right? We see nothing over here. So this model is definitely wrong. And what this means is, and we know from other experiments I don't have time to talk about now, is that the whole circuit has been reconfigured so that the sensory input combined with context is gated somewhere in the brain. We don't know where it is. It could be in dentate gyrus or even entorhinal cortex, who knows where. But the activity in CA3 is completely different in context 1 and context 2. So what really happens is there's a set of inputs that is associated with these. Think of these as axons from CA3 pyramidal neurons that are coding for sensory input in context 1, and there's a separate set of axons and cells that are coding for sensory input in context 2. And we've only potentiated these guys, so that's what's driving firing. So we have a better understanding of how the hippocampus is actually routing information, and it just raises tons of interesting questions, like where does this gating actually happen? And I think this is not a completely new concept, because the idea of circuit remapping and the hippocampal cortical system is something that's long been appreciated, but our ability to record from cells and do whole cell recording gives us a new way to begin to study this problem. Okay, yeah, that's it. This is what we're trying to do. We are ambitiously, I guess, trying to go all the way from molecules and cells through systems and up to behavior and cognition. Lots of people are trying to do this. It's hard, but this is, I think, what understanding is going to look like, is having computational models that help explain these connections all the way up. And maybe we'll never have a fully satisfactory understanding of the whole thing, but the more of these connections that I think we understand, the closer we are to being able to say we understand memory. And importantly, I think this is an understudied and underappreciated aspect of neuroscience, is what are the algorithms that systems implement that allow the animals to use cognition to drive intelligent behavior. I hope that we in my lab will spend more time on that in the coming years. Okay, thanks again to the people and the Mouse Light team. And I've got a few minutes for questions if you want to stick around.